Thank you. I would like to start by explaining what neanderthalization of brain organoids mean. And this is a new concept that we start working a couple years ago. So neanderthalization is a derivative of archaization. And basically what we are proposing here is the resurrecting of the extinct genetic variants that is found on archaic genomes in brain organoids. So it's a combination of paleogenomics, genome editing, and stem cell biology technology, mainly the brain organoid. So why we focus on the Neanderthals? Basically, I mean, I think we all know at this stage that humans have been sharing the world with extinct relatives, but the Neanderthals are the ones that we do have more rich fossil records. And when we look through time, there is really no evidence that the Neanderthals acquire arts, technology, and adaptation as we do today. So modern humans likely started in the same way, but in a very short period of time, we have developed such a rich history of arts, technology, and adaptation. We are basically everywhere. We are even in orbit at this stage. So what actually makes modern humans so dramatically distinct from the Neanderthals, which is very close to us, and even other species? So there is no other species in the world that's able to manipulate tools and technology in the way we do. So we postulate the following hypothesis that positive selection on specific genes might have resulted in the modern human brain. And our approach is very unique. We start with paleogenetics by aligning the genome of different modern populations with archaic genomes. And what we find out a couple years ago is that there is a catalog of 61 unique modern non-synonymous genetic variants in protein coding genes. These are the catalogs of mutations that distinguish us from the Neanderthals and other archaic genomes. So when you look into the 61 genes, there is one that calls the attention, and it's a gene called NOVA1, Neuro-oncological ventral antigen 1, which is a master regulator of alternative splicing that is highly expressed during the developing cortical genesis, which is exactly the model system that we have with brain organoids. So we thought that NOVA1, being a master regulator of gene expression, could control a cascade of gene expression changes between us and other archaic hominids. So what we have here is some analysis on the haplotype suggesting that the human-specific NOVA1 genetic difference, which is basically a change in isoleucine to valine amino acid change, in the second 3K homology domains in the NOVA1, these are the domains that are responsible for the alternative splicing. The NOVA1 actually interacts with the RNA, and we have cryo structure here. It doesn't show directly the protein RNA contact with that specific position. However, the analysis of NOVA1 is showing that the target RNA can bind simultaneously to these two domains that gets together to complete the genome, the splicing factor. The haplotype distribution is consistent with a relative recent emergence of this haplotype, followed by spread to fixation. So we measure this Tajima D here on the left of the human-specific haplotypes around all human-specific synonymous and non-synonymous difference. And we found that this is consistent with a purifying selection of the NOVA1 haplotype, which is quite unique. And I'll touch later on how this selection might actually happen. So once you find, the differences in those genes and even alterations in proteins changing amino acid, it's a long way from demonstrating that this actually affects neurodevelopment or how the brain develops. So the proposed work here is it's in the interface between all these genetic variants in neurobiology. So we need to test if the genetic variants are relevant. And here comes the brain organoid, which is our stem cell-derived neurotissue, also called in the media as a mini brain. We don't fully like this terminology, but each one of these white dots here will grow up to become one of these neurotissues. We can actually guide them to become different brain regions. In here you have cortical tissue. They end up with 2.5 million neurons in each one of these spheres, 5 million of cells in total, and they can survive for several years. Most impressive is the electrical activity that can be generated from these brain organoids. And to measure that in a non-invasive way, basically what we do, it is to plate some of these organoids on top of dishes containing electrodes. So these are multi-electrode arrays where the electrons are printed in the bottom of the dish and the organoid is on top. And all the time when there is an electrical activity passing by, it will be captured by the electrodes generating an activity map. The beauty of this technology is that you can perform a network analysis longitudinally. So we measure the evolution of these networks over time, over a full year period, comprising like prenatal and postnatal stages. And what we found is quite remarkable. We found that in the beginning, these organoids produce random spikes that starts to get organized when they are about 2 months of age. By 4 months, we start to see neural oscillations. This is suggesting that there are microcircuitries in the cortex being formed. By 6 months, these oscillations are quite stereotyped and coincide with the emergence of astrocytes, where we have an almost exponentially maturation of synaptogenesis. After that, we start to see a discrete population of GABAergic neurons becoming inhibitory neurons inside these organoids, inducing a more complex network that will increase the complexity as we mature these organoids over time. All these oscillatory events are dependent on both glutamatergic and GABAergic signaling, to suggesting that what we have here are all the basic components, cellular components of the human brain. But how do we show that this is exactly what happens in the human development? It's another story. So for that, we had to compare the emergence of these neural oscillations over time with EEG, or electroencephalogram, recorded from human brains start in prenatally. And for that, we use premature baby brains, where we place this EEG cap on them and we record over time. So we use a data set of different ages of premature baby brains to actually compare the potential ages of both the organoids and the EEGs using a machine learning algorithm. And that reveals that the organoids actually follow the same trajectory of complexity as the human brain does. Of course, the human brain has different brain regions that are not represented in the organoids. But nonetheless, for the features that can be compared, they actually follow the same trajectory as the human brain. So the idea would be, well, if you can make brain organoids from Neanderthals and compare to modern humans, that would be something quite useful. However, to generate brain organoids, you need to reprogram live cells, and there is no live cells coming from the fossil records. Even the brain does not fossilize, so we have to think about a different strategy. And that's where it comes this idea of Neanderthalizing human pluripotent stem cells to give rise to brain organoids carrying Neanderthal genetic variants. So we do that by genome editing using CRISPR-Cas9 to replace the modern version of the gene or the genetic variant for the ancestral or archaic genetic variant. So we are resurrecting extinct genetic variants back into a modern human background. And then we can compare how these brain organoids look like. And here is a summary of our data. On top, you have the organoids carrying the archaic version of Nova1, and in the bottom, the modern version of Nova1 that we all have. On the electrical activity, when we record this over time, we notice something quite interesting. The organoids carrying the archaic version of Nova-1 actually display an accelerated maturation compared to the modern humans. So modern humans are known to have a very slow neurodevelopment, even compared to other species. And so we are an outlier in terms of neurodevelopment. Our brains really take a time to develop. And this is clear with the analogy of a baby chimpanzee that can outsmart a human baby. So the maturation for a chimpanzee brain is much faster compared to the human neuronal maturation. However, over time, what we see is that a higher complexity coming from the modern version of the Nova-1 when we compare to the archaic version of the Nova-1. So there is an accelerated maturation at early stages, however, a lower complexity of these networks at later stages. And then again, this places the archaic version of Nova-1 as a factor that might contribute to the slow developmental timing in modern humans, which is quite interesting to know that. If that happens, it must have a strong selective pressure because we all have the modern version of Nova-1. But this is likely a sporadic mutation that was selected over time. So what could be the factor that selected the modern version of Nova-1? And we got some cues based on neuroprogenitor cells that has been exposed to lead. Lead has been one of the environmental stressors in these days, and we know that lead is bad for neurodevelopment. So several groups have been trying to understand how lead contamination might actually affect the development of the human brain. And this work in particularly find out that Nova-1 is the gene that responds to lead contamination. So that made us think that maybe Nova-1 is being manipulated or selected somehow by lead exposure. And it looks counterintuitive to imagine that lead contamination might do that. But our colleagues at Mount Sinai, this is Manish Arora and Renaud Johannes Boyan, they both show us that lead contamination is spread out during evolution in different primates. So this is not a recent event. It has been contaminating humans for the past 2 million years, humans and other primates. So how do they know that? They use a specific type of laser ablation on the tooth, the fossil tooth of these different species. And by ablating the enamel of the tooth, they release some of the components of the enamel, revealing eventual contaminations, including lead. And that's how they showed that the lead contamination was truly spread out through evolution, is not like a recent event from modern days. So if we were all contaminated by lead and Nova-1 is a key gene that responds to lead, it seems like an obvious experiment to expose our brain organoids, both carrying the archaic genome, as well as the archaic genetic variant, as well as the modern Nova-1 genetic variant to lead. So this work was done by Nina Sena, a postdoc in the lab, and when she exposed both type of organoids to lead, she found out that both of them react by expressing genes that are related to neurodevelopment. However, the archaic version of Nova-1, when in contact with lead, actually reduces the amount of FOXP2 positive cells. These are FOXP2 neurons that we can detect it using single cell levels. So consider the observed decrease in FOXP2 expression in the Nova-1 archaic cortical organoids, the further reduction of this gene expression following exposed to lead, and we decided to explore what kind of proteins might be creating this kind of toxicity or event. And that's when we turn into single-cell proteomics. And this was done by Aline Martins, a project scientist on the iScore Center here at UCSD. So Aline was able to isolate these neurons from the organoids and run the single cell proteomics, and she found out that most of the pathways are related to axon guidance, nervous system development, and the signaling by robo. So the alterations in the robo family identified through the single cell proteomics suggest that both robo might be involved in pathways that are related to the cortical thalamic axonal projections. And because of that, we decided to also create thalamic organoids. So Nina went on and generated these thalamic organoids and exposed them to lead. And again, upon exposed to lead, different from the cortical organoid, the thalamic organoid overexpress the expression of FOXP2. So both ROBO1 and FOXP2 has been previously associated with language and speech disorders, and FOXP2 plays a clear role in axonal outgrowth from the cortical thalamic projections. So the fact that they are both in the same complex, I'm showing here FOXP2 and OVA1, suggests that this complex is actually involved not only on the cortical thalamic projections, might be a specific pathway during the emergency of modern human language. We ask if it is possible that the NOVA1 would be like a gene or a variant that help us to develop such a complex language. And of course, I mean, the brain organoids doesn't have that complexity and is not linked to a body, so it's hard to study language development in a brain organoid. But while this study was being done, we were surprised by the work of Robert Darnell, who actually did the opposite. What he did was to swap the modern human version from the mouse allele, from the mouse genetic variant, in a transgenic model system. And he figured out that the NOVA1 substitution, the same that we did with the organoid, really changed the contribution to neurodevelopment, as he can start to see different vocalizations in these animal models, suggesting again that maybe this new NOVA1 substitution, the modern version that we all have, are probably contributing to the development of spoken language to this differential RNA splicing regulation during brain development. So in a way, it's an independent contribution, an independent validation of our hypothesis. So lead in language. So the in vitro system has shown that the ancient or the archaic NOVA1 gene has struggled against lead, likely disrupting FOXP2 positive neurons that are participating on these cortical thalamic projections. Would that give us like an edge for survival? Because if the version of modern humans that was selected allow us for a complex human language, that would be a dramatic advantage over all other hominids. So language is our superpower. It is what allows us to develop ideas, to plan the future. So we propose a very bold hypothesis suggesting that lead poisoning actually helps to drive human evolution by favoring the modern version of NOVA1 in accelerating the development of complex language. So it's a bold hypothesis. It's out there for other groups to help us prove or disprove that hypothesis. I'll end here by saying that there are a couple of take-home messages and limitations here. We find out that there are 61 genetic variants that distinguish modern humans from other archaic hominids. The archaization of brain organoids does not generate Neanderthal organoids, but rather a hybrid system. Remember that we are Neanderthalizing single genes one at a time, but the rest of the genome remains modern humans. So that's one limitation. The archaic version of NOVA1 definitely accelerates modern human development, but we don't know if there is epistatic contribution of other genes that might compensate for that factor. Remains to be seen. And the single nucleotide alteration in OVA1 was likely selected by lead exposure because of its protective effect on FOXP2 cortical thalamic neurons, allowing modern human language to flourish. So these are the messages that we have so far. We are definitely continuing on these experiments. And one of the things that I'm quite excited about, it is to start using a closed loop feedback between organoids and a robotic interface to start probing for memory and learning. So we previously showed that these organoids are able to, for example, manipulate a robot and teach the robot or remember commands to avoid obstacles. And the robot can use plasticity of these organoids to actually interact with different environments. And we do that as well as trying to map the circuitries that are inside of these organoids or participating in these organoids using high density multi-molecular arrays, such as the ones that are showing here on the right panel. So I'll finish here by thanking members of my lab, especially Nina Sena, who led the work, and our collaborations at the Manisha Arora lab at the Mount Sinai. And of course, the funding for this type of work. And here's my contact information. Thank you very much.